On the surface membrane of all living cells are complex protein structures called receptors. Similar to a lock, and HIV recreates the key. The two receptors on T cells on which the HIV can attach are the CD4 and CCR5. On the surface of the viral envelope are the proteins GP120 and GP41. Once GP120 attaches itself to CD4, its three proteins spread apart. This lets the GP41 protein, usually hid by the GP120 proteins, to become exposed and bind to CCR5. After attachment is completed, viral penetration allows the genetic core of the virus to be injected directly into the cell's cytoplasm. The viral envelope and the cell membrane are brought into direct contact and melt into each other. In order to release the viral RNA into the cell, the nucleocapsid needs to be partially dissolved through the process of uncoating. In order to reproduce, HIV's RNA is converted into a complementary DNA strand through a process called reverse transcription, using an enzyme called reverse transcriptase. Reverse transcriptase uses nucleotides to take a single-stranded viral RNA and transcribe it into a double-stranded DNA molecule. The new DNA contains instructions HIV needs to take over a T-cell's genetic machinery in order to.
The integration of the probe virus comes after the viral DNA is formed by reverse transcription. During integration, the double-stranded viral DNA is inserted into the host DNA using an enzyme called integrase. HIV integrase is an enzyme produced by retroviruses. Following integration of the proviral DNA, the protein RNA polymerase makes copies of the new genetic material and produces complementary DNA strand. Some of the new RNA strand acts as templates mRNA, while others are copies of the RNA genome. Next, HIV protease cuts newly formed HIV proteins into individual proteins. New individual proteins combine with copies of HIV's genetic information and create new mature HIV proteins in the host cell. In order for budding to occur, recoding must take place. GAG proteins recoat the nucleocapsid so the virus assembly process can be completed. Lastly, the virus pushes buds from the host cell that seal part of the host cell's envelope as they exit the cytoplasm. HIV glycoproteins cover the envelope, which allows the virus to bind to new host cells, being the CD4 protein and coreceptor.